Okay, so in this video, I'm going to take you through a 16S sequencing experiment, ultimately using nanopore sequencing. And this is an experiment affectionately termed the Shoe-ome. As you can see in front of me, I have some gym shoes, my gym shoes that I just went out to the car and collected. And what we're actually going to do is look at the microbes living on the bottom of a shoe. The thing that I like about this experiment is that it's personal, but not too personal. Every student is most likely wearing some form of footwear in the laboratory environment, so they are able to take something that's personal to them. But it's not as personal as other microbiome experiments could be, where we'd swipe an individual student, and then people might get freaked out by seeing that, of course, we're full of microbes, or they may find microbes and make wrong conclusions from that. So looking at the bottom of a shoe is a really nice alternative, and we find that it works well. In this particular experiment, the first part of it is quite simple. We're going to take a swab that's sterile and then also a solution, and use those two items in order to go ahead and sample the shoe. And then we're going to go ahead and use a kit-based prep. So today we happen to be using stuff from Norgine, and those item numbers and products will be on the website should you choose to use them. We don't endorse nor are we connected with them. And there are any number of methods and means that you could use in order to do the sampling and to do the DNA extraction. We just happen to use those currently. There are others that we haven't even tested yet that might work just as well, but that's what we happen to be using today. And we've used these with high school students, undergraduate students, and find that although some parts of it do have a number of steps, folks do really well, and we generally get pretty good results. So let's get started with the very, very first part, which is actually sampling the shoe.